But so everybody was happy for a break, so I got the chance from the chair to present my data at that movie. And when the lights went on again, there was quiet. Nobody said anything. And finally somebody said, well, if I would have seen that in cheap, I would have said, it's fake. And that was finally the consensus with which we left, that meeting. And that had demonstrated one thing, that this agent could jump a species barrier, because it was never outside of a hominin seen outside of a, but it jumped the species barrier. Now the question is obviously, if it can do mink, what else can it do? That was a question. And now I have a little bit my own favorite subject. I mean, I always considered mink as great monitors. I mean, farms, great monitors for the health of our feed supply, because mink carnivore is being fed everything we don't want to eat. The mink gets it, And so, if anything untoward is in our food supply, which does not show up very likely, the mink may be able to pick it up. And they did, because that agent must have come somewhere from their food supply. Now, by that, this was quite an interesting disease. It would have been probably this way. And I did one last thing. I made a gig transmission experiment on 250 mice, thinking I may hit a jackpot. And then I had to pack my things and go to Washington State to start my job at Washington State. And I turned over the whole thing to the able hands of Dick Marsh. And he and Bob Hanson then continued with this project here at the University of Wisconsin. They have shown, they have expanded our knowledge about pathogenesis, about the host range, and particularly they had discovered the experimental animal of choice, the golden hamster. Without the golden hamster, work on these agents would be very, very difficult. And so actually, we did, with this, I should actually go to close, but there is one thing more to be said, and that is, there is a certain Dr. Stanley Husinger, he discovered the prions and described that he said, unlike any other, unlike any other agent, it goes after they, it is actually, the prion is a folding mistake of a post-translational folding mistake of the peptide of the prion peptide. And that is, and this has the chance, if it is there, the mistake once done, the mistaken prion makes all the other ones follow suit, and then you have the disease. It's a precipitation of the prion peptides. And this had something to do with the mad cow disease, which came many, many years later, and then came the big question, what is the danger we have here? And the danger here, I can assure you, is, it can be relieved, it's not very much. I mean, we had about 7 or 8 outbreaks of TME as yet in Wisconsin, and none of them was followed by human disease. So whatever prions are circulating in our animal population in North America apparently have nothing to do with the BSE prion, which caused the outbreak in Great Britain. Thank you very much, I think.
What's going on in nature and really has permeated the whole arbovirus effort. And Bob was perfectly willing to look at new approaches in new areas, one of which I became involved in, but that was the question from the people in wildlife ecology that were working on snowshoe hares in Alberta, Canada, which have these hares have this population fluctuation. dramatically. And the question was, their disease involved, and of course the thing that interested me when I was a graduate student doing the field studies was how does that affect the dynamics of some arboviruses such as Western equine encephalitis, and then that led to studies, again influenced by Bob, on what were the environmental impacts that might affect the host-virus relationship. So it's kind of a transfer of his concern and interest in Newcastle disease that also applied to arthropod-borne viruses. Vesicular stomatitis was the other surprise. That's a cow's tongue, if you don't recognize it. And this is a major alley here with epithelium coming off. And even though it clinically looks like foot disease, it doesn't kill animals, but it certainly impacts production. And so it is a major problem, economic problem in South America. And the question was, how in the world did it get to Wisconsin and how is it transmitted? And Bob did some insect collecting by airplane with that, and that would be before these blood squashers got involved later. And it was interesting because then Lars moved on to the State Health Department of Georgia and continued work on VSV on an island called Ossabaw Island, which is endemic for SV. And I remember Bob showing me their plans for some study, where they were going to use sentinel swine and then catch insects and stuff, because the suspicion, although not proved, was that it was insect transmitted. And unfortunately, well, fortunately for Lars, but unfortunately for the study, Lars got called back to Ontario, to the OBC, and that study wasn't done until about 40 years later, when the group at the University of Georgia one day was being nailed it down. It was sandflies and translational transmission of the virus that was involved. Bob could have been by 40 years. Major problem in the American tropics in certain spots. And Lloyd Lauerman became really interested in developing a vaccine, and he tried it, and it worked pretty well. And he tried it in Panama, in an endemic area, but unfortunately it was never commercialized. We continued to do some work. I mean, this sort of, we get onto a theme and keep going on work subsequently that we did in Colombia in South America, where there are areas that VSV is also a problem. And this is how Bob spins off ideas. Jeff Letchworth came onto the faculty, was interested in VSV, and he, with one of the Colombian graduate students, developed or tested out hypothesis that maybe there was persistent infection, and that might account for some of the cases that popped up. And they developed a hamster model that showed that. Due to we had a large group of students from Colombia, as you know, and one of the big problems there is Venezuela encephalitis periodically has tremendous outbreaks in horses, 80% case fatality rates, and it doesn't kill very many people, but it fills up your hospitals. So a major zoonotic disease, and that interest then led us to do some work in Colombia with looking at following up on some field observations that maybe black flies are involved as vectors. The conclusion was, well, maybe a few of them are, but a lot of them aren't. Also appeared in Wisconsin here, one of these arboviruses that came to us was epizootic hemorrhagic disease of deer, which is capable of causing major die-offs in deer in the country from the southeast all the way up into the Dakotas. And that led Bob, again, Lars was involved in that early on, and then later Dan Trainer looked at epizootic hemorrhagic disease and wood tongue, looking at pathogenesis of that particular virus disease. It's transmitted by no-see-em, pigeons. And it's a serious, if you're here and you're in big trouble, if you get it. And this is still a problem, but some of the early work, again, is a good illustration of a problem that appears, have a look at it, figure out what's going on, and then for further work, sort of spin it off to those who are interested in doing it, which he did with Dan. And Blue Tongue sort of was on our minds, and that led to a large collaborative effort that was spearheaded by Jane Homan, looking at the occurrence of blue tongue in Central America, because there were certain animal regulations that said if Guatemala had it, you couldn't ship animals to Costa Rica or cross international borders there in these very small countries. And the question was, well, is blue tongue really a problem there? And Jane got all of the Central American veterinary and some of the Caribbean islands veterinary services dancing together the baseline of Costa Rica that showed that they were all served with the same viral soup. And also that it's a dynamic situation, that a blue tongue strain appeared that had never been seen in the Americas before, only been seen in Africa. And the question is, how did it get across the Atlantic? The hypothesis is blown insect. Further work on blue tongue by Kevin Schultz and Kristen Bernard had a look at the structure of the virus and how that influenced pathogenesis. So it's, again, this whole idea of you start out with a problem in the field, a real world problem, not something you invent, and then you follow it through to find out exactly how things work in nature. And there was a question of, most of these arboviruses involve wild animals, either birds or mammals, and they usually don't have an adverse effect. The animals don't complain. They do well in school. And the question was, well, do these animals really pay a price for it? And this is one of the themes, one of the areas that Bob was interested in and that he encouraged us to go ahead. So we put an interdisciplinary team together and had a look at Venezuelan equinoencephalitis virus strain. This is a vaccine strain that's kind of mild because it's used in horses and people. And do the animals pay a price for doing it? And the answer is, if it's combined with sublethal toxicants, environmental pollutants, that the animals do pay a price in terms of reduced reproduction. And in wildlife, this is really hard to measure, especially in the wild. One of the biggies that has led to studies that have gone on for 40 years is the appearance of lacrosse encephalitis virus. In 1960, a child from Minnesota got sick, went to the Gunderson Clinic in La Crosse and died. And brain tissue from the autopsy was sent to the state hygiene lab, and it sat there for about 4 years until Wayne Thompson, who graduated from his PhD from Vet Science, took that job and isolated California encephalitis virus for the first time ever. And then the question, of course, is, okay, what's going on? Where's the virus hanging out? How is it transmitted? Closely related to an insect, mosquito-transmitted virus in California. So it's part of a group. And so it was a problem also in southwestern Wisconsin, in the Griffiths area. So what is it out there that's causing this virus to persist and being transmitted? And so a team was formed, and Bob was very active in it. And I think this characterizes the way Bob operated a lot of times. He wasn't sort of, if you asked the group who was the leader, they wouldn't have said it was Bob. But if you asked him who provided most or a lot of the imaginative input, everybody would have said, yes, Bob. As I became part of that group, and it was really marvelous to see Bob operate quietly, no ego, great suggestions, constructive, friendly criticisms. It kept everything going. And Gene DeFollier, the entomologist in the Russell Lab, students were running back and forth with mosquitoes and stuff to the vet science lab, the Bob's lab, to do the virus work, because they didn't have that capability. So they found the Aedes tricereatus, the white meat, bait-biting mosquito that likes to feed on mammals, including people. And that mosquito had the virus. Then the question, well, where are they getting the virus from? And so then wildlife group, mosquitoes, and from my lab, when I came back from Columbia, started asking that question. It turns out chipmunks are pretty good hosts, squirrels are too, and so are foxes. There are some effects on pregnancy, but the big question then was, okay, well, where does the virus spend the winter? Because in mid-October, when we get the first killing frosts, the adult mosquitoes, the adult Aedes tricereatus, are all dead. And so where's the virus hanging out so that it gets started again the following summer? And we had a graduate student who was actually from Thailand, who I worked with when I worked in Bangkok, came over to do graduate work, and we had him doing some work with chipmunks, thinking that chipmunks get infected, they hibernate, they come out in the spring and take the virus with them. Well, we did work in the Biotron. In that case, Dave, the Biotron actually worked. But it was to give up on an attractive hypothesis, but the chipmunks cleared their infections. They never carried it really into hibernation and came out with it. So, of course, Omzak was so frustrated that the main field study area was the Pinnacle Farm, the Hanson Farm in Iowa County. And it's really great to have your major study area in friendly hands. So he was crawling with graduate students. And Omzak, just this time of year in April, he said, I'm going to go out and look at larvae, because what happens is you accumulate water in this base of tree hole, and it's like a little basin, and the mosquitoes love it. That's where they breed. And so he sucked up a bunch of larvae, brought them back to the lab, breathed them out, and got virus. And I think the thing that was interesting was, at that particular point in time, the conventional wisdom was that mosquitoes could not transmit their virus through the eggs. And so Bob told Omzak, well, yeah, go ahead and try it, but don't be discouraged if it's negative. But he didn't tell Omzak, no, everybody knows that doesn't happen. Fortunately, and I told Omzak essentially the same way to try it. I knew we needed to get out of the Biotron and out to get some sunshine and on his pineal gland and feel good. And they were positive, and that was the first convincing demonstration that the virus, that arthropod virus could, in mosquitoes, could go into eggs and into that generation. And that was revolutionary. That got published in Science. So there was a good integrated group. This is an interesting group. It was interesting to watch the social dynamics in that sometimes we had turbulent times in the arbovirus group when we get together. And the soothing, calming influence was Bob that sort of kept us on track. So a continuation of these lines that grow out of these surprises is then there were further studies on the virus-mosquito relationships and why are some mosquitoes susceptible and other mosquitoes are not. And although Bob wasn't in a supervisory position in that research, he was a participant. And this is the anatomy of the lacrosse virus in the California groups, and there are 3 segments in the genome. And at that time, there was getting to be an interest in whether these things could read a sort, recombine. And so some of the graduate students in our group pursued that, and also wondering why in the same woodlots there were two related viruses circulating, and yet they seem to be separated in different cycles of transmission, with Jamestown Canyon involving deer and the Hibernates mosquito, and La Crosse virus in its small rodent hosts and Aedes triceriotis, and yet they always seem to be separated. And the question then was, well, if one were to infect a cell with both viruses, would they swap these segments and you get new viruses out? And the answer is, if you do it in cell cultures, yeah, they can do that. So biologically, it showed that they were capable of doing that. But then you don't worry about cell cultures, you worry about what's happening in nature. I mean, that's the answer to this so what. Eventually, and the answer is the Asian tiger mosquito, which has been introduced into the Americas, including the United States, which is susceptible to both viruses. It has receptors to make that possible. And if you infect them, you get all 6 new viruses out of them. So, well, should we worry about that on Pinnacle Farm? Is that farm still in the Hanson family? Yeah, it is, isn't it? Yeah, well, is it coming your way?
I haven't gotten sick.
Okay, well, you may have that opportunity. This a bit outdated. This mosquito, as temperatures have increased, the distribution of this mosquito has changed. And now you can see it's as far as the Chicago suburbs. So it's not that far to Iowa County. And if you get it, then you may get Jamestown Canyon and La Crosse virus and their progeny, their new ones, coming out as something new, as a new product of the Pinnacle Farm. So, well, to continue the tradition and the environment that permits this kind of exploratory research to go on is an example of Florio, who's kind of my albovirus replacement when I retired. And it's dengue fever, and so what in the world is a veterinarian doing studying dengue fever, which is a human disease? Well, the spirit in the department has always been, it doesn't matter. If it's an interesting problem, go for it. And the fact that this isn't restricted to physicians and schools of public health, there's nothing wrong with a faculty member from the Department of Pathobiological Sciences in the veterinary school having a look at it. He's been working on a dengue vaccine and doing field tests in Columbia, where he's from, and has strengthened the Columbian-UW connection. And although Bob had no contact with Tony Goldberg, Tony has done beautiful work on the ecology of Western Alberta and birds in the Chicago suburbs. And if Bob were still around, he'd love it. But it's the spirit that was established that makes all this possible. And so the question ultimately is, how are we doing? Are we ready for the next surprise? Will we respond to it in as organized and imaginative way as Bob had done in the past? Would we put field and lab studies together? Would we work as teams interdisciplinarily? And I think the answer is yes. I think that legacy is still very much present and alive in on campus generally. And so I think the legacy that Bob established, that popped up by accident in 1952, is still alive and well, and I look forward to the next horrible surprises that pop up and how we might actually get at them and find out what's going on. Thank you.
That's it. I know we've been sitting for a while, so I'll be brief. Only a few minutes for you. I want you to understand most, if not all of you in this room have had a privilege that I've ever had. For some, Dr. Hanson was a stimulus and encouragement to your studies. For others, he was a friend and mentor. For my mother and uncle, he was a father to come home to. I have an unattainable envy of all of you, because for me, he was a grandfather that I will never have a chance to meet. My mother would tell me stories, though, of his innate wisdom his understanding of the connection of all living things. And it's this wonderful connection that allows, wondrous ecosystems to flourish. However, it also enables disease's ability to spread rapidly beyond control. And instead of a reactive approach forced onto us from dire situations, Dr. Hanson wanted a proactive approach. He's trying to continue research for a better understanding of diseases that we still face today. It is in the simplistic difference of words, but drastic difference in implementation of reactive to proactive that made Dr. Hanson a visionary in the field of science and health. We are not here today only for his academic success. We are here because of his compassion. An open-door philosophy is a characteristic that's esteemed among students, such as myself. But Dr. Hanson did not have an open-door philosophy. He had an open-life philosophy. Whether you were a fellow professor, a coworker, a graduate student, it did not matter. All were invited to the family farm just outside Madison, Pinnacle Farms. It was in this nature-rich getaway that people could hike, people could have picnics, people could have cookouts. But above all, it was something more than that. There was a place to explore. There was a place to cultivate new ideas and new approaches. See, even away from academia, he surrounded himself in an environment that aided in his cognition. I would like to share that, once again, I am graduating with a few weeks from this university. And although I have that unattainable envy of you all, we also share something very special. Bob nurtured his students to go where his interests took them, whether by example or by mentorship. He led us to achieve further. This inspiration is what we share, and now it's become so much more. See, up until this point, he's only been stories told to me, and he has only been a picture of my bed. Now, with the overwhelming honor of this building, his legacy has become tangible. It is unquestionable through my academic career. He's been an inspiration to me, and it's my hope and my belief that through this legacy and this building, he becomes an inspiration not only for myself and for you, but for the students who will continue to come to this university for many years. Thank you.
Close with some comments from Bob's son, Alan.
What you're seeing right now is the slideshow that I put together as the veterinary pictures, or the pictures that we had in his colleagues. I don't quite know exactly, but at the end of this was taken, and there's a discussion of why, what it was. And I went on and researched and found out that she was interested in childhood diseases. And that's like what we know is there. This is his office and his students. This is the veterinary school.
Veterinary department.
Department picture.
That was the day when you celebrated his National Academy of Science.
Yeah. Yeah.
Jose. Yeah.
Okay. But this is on top of the pinnacle. This is the heading to the article of the Bloom series. It started in 1940, and this is like the article that came out every Friday. And this was to be the cover of his book, which he was making about the Pinnacle Farm, which is about the ecology of blooming animals and everything. It was to be an almanac. And then it was like the Earth Day in a cycle as it would breathe and different things animals do. And this was one thing he wrote for the introduction to it. And he's talking about, I don't think anybody's ever seen this. He's at home. This was like a plan for it. Like what I'm thinking that can be done with it now. This is a plan, like you got the different habitats where the different things are happening. This is where an example of like the first week, they're divided into weeks in the circle. This is like at my website where I put them up so you could view them with a possible way. And so we finished. We'll just recycle. Basically, the Bloom series, he had a group of friends and they wanted to save the Apostle Islands, and that's where that started. And those people just, when he went away in the 40s, went into the Army, they kept publishing that for about 40 years. And it was a dedicated group of non-collective people about it that it brought together. something else. No. This was his trip to Norway, where he met relatives. This basically was a place at where there's a nuclear power plant now. And we went there on a vacation. We didn't have a car, and it was 1957, and we went by bus. Oh, these are the people that were in the Plum Island. Oh my! This is Hitchner. He wrote a paper in one of the articles. Family there. This is the sorghum, or basically it's broom corn. And that his idea with that is he came up with a project where they were going to have Newcastle disease. And what they were going to do in Africa was have a vaccine so that he was going to feed Africa that if you vaccinated the chickens, they wouldn't all die. And so you would take the sorghum grain, feed it to the chickens because it's high in protein. The chickens, because most of the grain would be eaten by rats, like 80% of it, with the graduate students from Nigeria, and then they would be able to feed themselves. They made a patent, and I don't know whatever became of it. I have a website, which is lhanson.com, where I put all this stuff up there so people can view it. And most of the things I put in there were PDF files, so you can copy them. I'm going to put more and more stuff up there. So like everybody, all the papers, I started putting up all the papers that he'd written because I discovered that some of them were going missing and we couldn't find some of them. And my mother found a bunch of books. She thought she'd collected everything. Some of it isn't too good. Some of it's pretty good. Like all your stuff that you wrote with him is there. Will be there. And I think it's something digitally the university could do. It's like have an archive of stuff. That's pretty much what I have to say.
Al Hanson. Website, Al Hanson, one word or two.
That's all small case, one word.com. You're going to wind up going to a car dealership with an E. And it's going to be in HTML7, and I've set it up so it's tricky, like everything's a link. It's all objects. And you need a Chrome browser, a Google Chrome browser, or it won't run. So it's kind of experimental if it doesn't work.
That's pretty nice. Very nice.
Just a few minutes here until we all wander down the street, because we do want to be down there in time. Comments, who's here in attendance today. We have many people, family, friends, colleagues in attendance. Is there anybody else who'd like to say anything here to the group? Sure, go ahead.
I'd just like to give my appreciation as a nephew, seeing totally that when he was with family, he was just family, right? You talk about his interest in science and stuff like that, but it was mostly when he was with his brothers, they were brothers. So it really gave us a vision of what he was that we just never saw. So I appreciate all you've done.
Our pleasure. Anybody else?
I would like to say something. Just to extend a little bit, one of the things I really appreciated about being a grad student in Wisconsin is the help from all the other grad students. The part of that friendship that Jane went and got up at 4 o'clock in the morning and go to sled ducks that we were tracked and taught me everything I know about Newcastle disease. So in terms of the labs, the thing. But the other thing I wanted to, we all know that we have extended this to South America and to many other countries, but also to Antarctica, because that's where I went when I left Wisconsin. Wisconsin winter wasn't bad enough. Good.
And I have a question for you and Jane and maybe Sue Island. When the group was sampling ducks out on the Mississippi, and there was a tavern that had a big sign in front that said smoked carp,
Yes.
and somebody of this group interchanged the two little letters.
That's Las Vegas. What's the
We're not telling.
Was that an analysis or was that?
Find out.
How do you want me?
I'd like to make a comment. Sure. Dr. Hanson was on my PhD committee. I received my PhD under Barney East-based supervision. But since I was working on Newcastle, I had a lot of interaction with Dr. Hanson. And what was interesting about him is we would go over, we graduate students, to the Pinewood room to eat lunch many days. And Dr. Hanson or Dr. Berman would be, maybe together, would be sitting out in the Pinewood room. And they'd motion us over. So here are these distinguished professors asking their graduate students to come and sit and enjoy lunch with them. And it was just a wonderful relationship. And we talked research. He wanted to know what we were doing and, what was going on. And he was on my committee, along with Dr. Berman and Dr. Walker. And it came examination time. And I had been told that one reason you have a final examination when you're getting your PhD is to remind you that you don't know everything and you've got to keep learning, okay? So I had that in the back of my head and we went into the room and the exam proceeded quite nicely. And Dr. Hanson looked at me and I hadn't been reading Newsweek and Time and I was really boning trying to get the research work done. Dr. Hanson looked at me and said, Dr. Beard, would you comment on what you think the potential epidemiological impact is going to be with the introduction of the manatee into our waterways?
And did you say no? That was the answer.
I was smart enough not to try to bull my way through that. And I had been told, don't be afraid to say I don't know, but do not try to smoke and mirror that commitment. And that flew up in my face, and I said, Dr. Hanson, I've never heard of a manatee. And bless his heart, he was right. These things had been introduced, they were mammals, and introduced into the Florida waterways, and they were all over the freshwater part of Florida. But every time I think of my dear beloved Dr. Hanson, I think of the mammal.
The advice was offered to say, no, I don't know. Marty?
I just wanted to say that I think in the program it said something about tours of the Hanson building. There's no tour as such. Feel free just to wander around the building, look at the places where you work, and so there's no tour as such, but it's open to wander around and to wherever you like.
Jay, I'm sorry, I skipped over you.
Yeah, I was just going to say a couple of things. The first is, it came to me this morning that it's actually 27 years since Bob died.
Wow.
And that meant that he operated in a world before computers. In fact, I remember coming back here as a postdoc and kind of grumbling that I didn't have a computer, and he arrived in the doorway of my office very proud that he had found me what he thought was a computer. It turned out to be a dumb terminal from a Wang, but that's okay. He thought he found me a computer. But the point I'm getting at is that all of those papers were written by hand or were typed. I mean, we're so used now to being able to edit and re-edit and re-edit and delete and retype everything. And it's just kind of a bit mind-blowing to think that all of that was done without a computer. The other thing that relates to that is I keep thinking, pretty much on a daily basis when I pop open ProMed, what would the combination of Bob Hanson and ProMed have been? You know how excited he was when he got some piece of information and he would rush down the hall like a sort of hyperactive sandhill crane and grab the nearest person and start, expounding on the latest idea he had, whether it was a student or a stew bat in the glassware kitchen. Somebody had to listen. What would he do if every morning, Tom, he opened your ProMed? And wouldn't he be so excited about how you're getting the word out by being involved in that kind of activity? And then just the other thing, I was commenting to Chuck a few minutes ago that being here is like being in a big family gathering. Right. Except we actually probably have much more in common than a family gathering of this size. So it's just great to have this kind of get together. And thank you for putting it all together.
I'm sorry? Yeah.
One of the things I noted on one of the slides Tom had here at the end, he had a number of names and authors, and one of them was Baby. And that's Barry Baby. And to show you again, there's a circle here and a familiar sort of thing. Barry Baby's daughter, Lyric, did her PhD with Bruce Christensen a number of years ago, and she's rejoining she's joining the School of Veterinary Medicine faculty in August. So again, we get these circles of people, and the web that's begun years ago continues to operate for bringing people back here. Other people have comments?
Yes. When I came from Colombia, and he decided to be my advisor after he found me at the hall looking at the different places to see where I was, because I didn't have any information when I came here who was going to be my major advisor. So I was standing there for a long time, and he passed 3 times. He used to come to the secretariat from time to time. At the third trip, he told me, well, may I help you? Well, thank you. I'm just coming here from Colombia, and I'd like to have some orientation. Okay, come to my office. So he took me to his office and asked me about what I was planning. Probably he knew that I was coming, but it was not quite clear who he was going to be my advisor. So I explained him my situation, that I was working on diagnostic activities. I had a lot of problems to isolate viruses, and he said, okay, I can help you to give some orientation. So I'm going to be your advisor for 1 month, and then you will decide, we will decide who is going to be your major advisor. But in the meantime, you are going to take Dr. Pratt and Deming course, my Robert course. So we were with Charlie, with Dick, and David. That was a terrible course. That certainly helped me to learn everything. After that course, I didn't have any problem. But in order to advance in my research, said, okay, you have problems isolating viruses because they don't bring the samples properly refrigerated, or they have to come for a long place. So let's do an experiment. And the experiment was how to recover viruses from chickens that have been under different temperatures. So I have 1 set of chickens in the cold room, 1 in the freezer, and 1, he said, you have to have them at the temperature that you are going to you have in the tropics. So I have to set an experiment with chickens that I have to put in a box with a 70 degrees temperature, and I have to take samples from each one of them, different samples that I had. So everything was going very nicely. I had to develop a technique how to get liquid from the gut in order to get these samples and everything. But the problem appeared that the experiment was supposed to last 72 hours. And the last 24 hours of the chickens and 70 became so all spoiled and rotten and smelly. I was able to take samples until 48 hours. But then, when I arrived the next day, the administrator, I don't remember the name of the administrator, he was really very mad, upset because the whole building was smelly. So he threw away my chickens.
There was this crisis of beef in Argentina. This is during the Kennedy administration. So my dad got to go to the committee in the White House, and they were thinking this is going to be big money, and it was kind of like crisis today. So they asked him as a specialist, how much it would cost, and he said $52,000 in 1 good man, and I'll be on the faculty. Kennedy just looked at him. So then after that, he met with him because he was interested in him. So then he told him about the Apostle Islands. His this dream as a kid of making it a national park. So Kennedy decided he had to go there. So that's why the airport and all that. And that's
But just to
like
finish-
all of his things, that's probably the Rosalind Carter thing is the reason that picture is that he'd come up with something that wasn't millions of dollars, but a very simple solution.
Solution and form.
But just to finish the experiment, of course, I had samples until 48 hours, so we were able to test all of them. And we found that the virus could survive 19 hours under 70 degrees temperature. That I would say, well, if I process the sample properly, I could recover virus even if the samples were rotten. So, I thought, the next weeks I thought, what kind of a silly experiment I was involved than the problem that I caused. But at the end, we had very, very, very good results. And it was because I asked him to give me orientation and training to go back to my country to help in the establishment of the diagnosis services and of the animal health programs. And certainly the influence that he had, it was excellent, because when I went back, fortunately there was a crisis and they were reorganizing all the veterinary services, and I had to help to organize with the training that I had. And as Olga mentioned, at that time we started the foot and mouth disease program eradication that fortunately at this time Colombia is free of the disease. And then I follow up in the Pan American Foot and Mouth Disease Center, where he attended all these scientific committees, and the orientation was to change the strategy, not to do all the same full-time vaccination or to the whole country, but select strategic areas where the disease could remain or be the source of the virus. With that strategy that we developed in South America, almost all South America is free of foot and mouth disease now. And he was in the scientific committee of the Pan American Foot and Mouth Disease Center for almost 10 or 15 years when we were developing these strategies.
Well, with that, I think probably a good time to draw things to a close.